Thank you so much, Dr. Mansui. Hello, everybody. As Dr. Mansui said, I'm not a scientist. I'm a funder and advocate for research into heritable effects of germ cell exposures. Because I'm a little bit unusual here at this wonderful symposium, I just want to provide a few slides for context. What we do with our fund is we focus on what I call big doses of toxic stuff. In the world of epigenetics and epigenetic inheritance, there's a lot of emphasis on what I think of as the lifestyle or environmental exposures, and those are very important. But what we do is really focus on high dose things like drugs or tobacco. And when you think about it, it's these types of exposures that have, at least so far, provided the strongest proof, in my opinion, for non-genetic inheritance happening in humans. And that's particularly true with respect to the old pregnancy drug, diethylstilbestrol. My concern about the adverse impacts of germ cell exposure stems from my own history. My husband and I have 3 very beautiful children. Our younger son and our daughter are both severely disabled with nonverbal idiopathic forms of autism. As you may be able to tell from these pictures, they're really quite lovely. They're very healthy, but their IQs are in the 30s. They cannot talk, they cannot read, they cannot write, they cannot take care of even their basic hygiene. They've never played with a toy. They lack capacity for symbolic thought and basically lack capacity for almost all forms of learning. Now, clinicians have found no risk factors to explain these incredibly profound disorders. There were no risks in my pregnancies, no mental disorders at my family tree or at my husband's family tree, and so far no mutations that we can find. But about 8 years ago, something very bizarre happened, and I just happened upon some old records that showed that when I was in utero, my mother had been given very, very heavy doses of synthetic steroid hormones that at the time, I was born in 1965, were thought to prevent miscarriage. Now, they didn't prevent miscarriage, we know that now, but that didn't stop some clinics, especially like in West LA, where I was born, from using these novel chemicals in obstetrics. So I couldn't help but think that perhaps this fake hormone protocol had done something, I couldn't imagine what, to my developing tiny eggs, and that somehow that later dysregulated the cortical development of my children. It's really important to address something now, which is that in the media, it's often said, often portrayed, that the autism surge is just an artifact of increasing awareness or broadening diagnosis or shifting, categories. That is emphatically not true. There's overwhelming evidence that we have experienced a dramatic and very, very problematic surge in autism over the last 3 decades. And here's some data from my home state of California. You can see that the surge of prevalence of autism in our developmental disability system, these are only the more severe cases, has grown considerably. This is, again, prevalence. This is not caseload, and this is only those live-born in our state. And this is despite the categorization of autism in our developmental services system becoming more stringent, not broader, over time. What we're seeing now in California is that fully 2% of all males born in our state are now ending up in our developmental services system as autism cases, and this is a disaster for which there is no precedent whatsoever. Now, at the same time, research has shown autism to be a highly heritable disorder, although one, frankly, with very few identified genetic roots. Some geneticists speculate that common variants could explain this vast missing heritability. But I cannot help but wonder, instead, about a non-genetic inheritance story here, and if so, if germline toxicants could possibly be part of it. One thing I have the liberty to do as a non-scientist is just talk up autism parents, right? I don't have an IRB, lording over me. I could do whatever I want. And so I do that. I survey parents. I talk to them when I'm at parties. I sometimes have friends, and I'll call them up and ask them questions. And I've probably conducted more than 200 of these interviews over time. And what surprised me in these interviews was something I wasn't expecting at all. It wasn't even on my radar. But one thing that surfaced over and over again was the early surgery of an autism parent. So it got me thinking about agents of general anesthesia, which I'll call GA for short. Now, of course, GA must largely be very innocuous to germline. Otherwise, all of our kids would have neurodevelopmental problems, and that's clearly not the case. But I started thinking about if there could be exceptions to this general rule. Now, I have lots and lots of stories, many dozens of stories I can share with you. Obviously, I don't have time. But here are two examples of very early exposure to general anesthesia. Both of these moms are local friends of mine. They both have 2 sons with idiopathic autism. The mom on the left has 2 beautiful boys who are not twins, by the way, who are both significantly affected. And the mom on the right has 2 sons, one of whom is extremely severe, that's Muhammad right here, and another who is high functioning. But they have something in common in that they both of these moms underwent surgery very, very early in life as neonates. The mom on the left was born in Nicaragua, and she had a surgery for removal of a benign tumor on her spine and another for a hernia repair. On the right, this mom had undergone surgery at 2 days old for adenoid removal. So as a rough control group, I asked, is there autism anywhere else in your family? Any other kids, any cousins, upstairs, down, anywhere, downstream, upstream? And there was no other autism in these families, and this was a pattern I saw again and again in families. Now, to be fair, I just want to say something about general anesthesia. Timing might not be the only factor here. General anesthetic gases have to be used in unusually high concentrations in neonates and young children. Also, to be fair, general anesthesia in the past often wasn't used in neonatal surgeries. They just used muscle relaxants and nitrous oxide. Of course, we don't have access to any of the records. We don't know for sure what was used. Now, here's another time frame or another example that stood out to me. These are 2 friends of mine, again, local in the San Francisco Bay Area. They both have sons with extremely severe forms of autism. So to give you a sense of how severe they are, I'm just going to show you a 10-second clip of my friend's son, Pablo, here.
Aiden, you just stay calm. Sit all the way.
We don't need the audio. So what you saw there is my friend trying to feed Pablo some breakfast, and he's so self-injurious he has to wear arm limiters so he doesn't bash himself in the head, but nevertheless he finds ways to injure himself constantly. And on the right, my friend's son Luke is somewhat similar, but he also has some added physical abnormalities and detected defects in his corpus callosum. So I'd known both of these moms, these families, for years. And I know they had no risk factors in their backgrounds that would hint at anything like these extreme outcomes. But very recently, I had discovered in just casual conversations with them that both of their husbands had suffered gunshot wounds in their youths. And so here you can see my friend over there. Her husband had a gunshot wound to the eye. It was actually a BB gun. And he had 5 reconstructive surgeries following that accident. My friend here, her husband had a gunshot wound to the leg, a hunting accident up in Maine, and he had 4 surgeries at the age of 16. So perhaps it's entirely coincidental that in both cases the fathers had these highly unusual histories. But me being me, I couldn't help but ask if there was something about the repeated GA exposure that could have damaged some of their spermatogonial stem cells in some fashion. Now, I share all these examples not because I think they alone prove anything, of course they do not, but because they are representative of similar stories that raise, I think, crucial questions about potentially catastrophic heritable dimensions of this exposure, which among all the neurotoxic exposures we humans suffer is fairly unique in its intensity. Now, no one can say for sure if it's cause or a coincidence because there has not been a single study on the heritable impacts of general anesthesia in humans. But do we know anything? And yes, we actually do know some things despite this omission. So as background, volatile anesthetic gases, which there are other forms of anesthesia as well, but I'm focusing on those here. They're small lipophilic molecules that passively diffuse through cells and germ cells and can enter the nucleus. They're very prevalent, up to 75,000 pregnant women per year, 6 million children, of whom 1.5 million are infants undergo, I think this was just inpatient surgery, 48 million surgical inpatient procedures a year. So which of these, and as you can see, they were introduced with halothane in the late 1950s. Now sevoflurane is the more popular of the gases. Halothane is banned in the US now for various issues around toxicity and flammability. On a fairly simple level, we know that GA can damage the integrity of germ cells, and the endpoints include abnormal morphology, abnormal concentration, motility, and abnormal effects during spermatogenesis. The pathologies seem to be related to GA's disruption of normal reproductive hormones. They are also genotoxic. They aren't known to be mutagenic, by the way, but they are known to be genotoxic, and that was seen primarily in somatic cells. And also in oocytes, there's been very little, as usual, the oocytes are often overlooked, very little research, but there has been at least some injury demonstrated. So there's quite a bit of somatic toxicity literature, mostly, of course, relating to neuronal development. That results from early life GA exposure. This is a source of growing concern in the anesthesia community. And so what we see in the literature, both in the experimental models and also in the human studies, is that there is at least some degree of learning impairment and behavioral abnormality when there is an early life GA exposure. And the mechanisms include apoptosis, impairments in synaptogenesis, in neuronal migration, and disruption in the GABAergic system, among others. So what is seen in this literature is that GA is a powerful modulator of chromatin remodeling. And what I ask is, if it's having the same effect on germ cells, could this explain maybe some of the autisms? And what you see in autism, post-mortem autism brains, again, this has nothing to do with GA, is failure of cortical neural migration, cytoskeleton impairments, synaptogenic impairments, neuron loss, and disruption of the GABAergic system. So there is some mapping of what we see in autism to what we see in these early GA exposures. I'm not saying there's a cause here. I'm just remarking on that similarity. Now, there are only 4 mammal studies looking at heritable effects of GA exposures. 2 of them were done in the 1980s in a lab at NYU. They looked at the developmental effects of early GA exposures to halothane and enflurane. In a small experiment, they exposed gestating mice to halothane and observed that the progeny of the exposed fetal eggs exhibited impaired learning. The researchers thought this result suggested, quote, that the anesthetic agent may have caused a genetic aberration in the fetal eggs. In a subsequent experiment, the same lab exposed sires to enflurane and again found impaired learning in the progeny. The authors stated, and again I quote, it seemed likely that spermatogenic changes caused by the gas were associated with some sort of genetic alterations, unquote, in the germline that affected the progeny's brain development. Now, these novel and alarming observations fell into the abyss for 34 years. No follow-up whatsoever, nobody asking any questions. We can't find anything in the literature relating to heritable effects of GA for 34 years. Perhaps because people couldn't conceive of a mechanism. When finally, just last year, another researcher published on this phenomenon, the Martignac lab at University of Florida. and they did 2 rat studies, one of which was published last year, one of which should be published like this week or next week. But they found similar results. The first study exposed neonate rats to subclinical doses of sevoflurane. They found male but not female progeny exhibited learning and behavioral impairments, and this time they were able to scratch at the surface of a mechanism, and they found the behaviors related to epigenetic modifications of a gene important for GABAergic function. It's possible that other pathways were involved, but it was a preliminary study. The second study, that's not yet published, exposed young adult rats to sevoflurane, and again, the male progeny of the exposed germ cells exhibited these impairments, also connected to the modification of the gene as found in the progeny brain and the germ cells of the exposed rats. So while there are only 4 studies on this phenomenon, they all seem to point in a similar direction. So from an experimental level, we can see that GA probably can exert adverse heritable impacts, particularly in male progeny. So my summary is this. We've seen a dramatic surge in autism, which is a highly heritable disorder, but so far genetics has not explained this high heritability. And unfortunately, the mainstream of autism research doesn't even consider the possibility of non-genetic inheritance. So our fund tries to fill this gap with pilot projects and research advocacy. Heritable impacts of GA has emerged, surprisingly, as a crucial question. We need to somehow get studies going in humans. I'm working on that right now. I've funded 2, hopefully a few more on the way. As I said, the mammal studies are very concerning. The mechanisms seen so far are epigenetic, and more research is desperately needed. I have no lab to thank, but I do want to thank my friends at the Environmental Mutagenesis and Genomics Society. And I know a lot of you haven't heard about EMGS, but this society launched 50 years ago out of a concern that the glut of post-war drugs and chemicals could damage our germ cells' chemistry of heritability. And it was a vision very far ahead of our time, but it's a vision that still has not been truly realized. I also want to thank the families that shared their exposure histories with me, and if you want to reach me, there I am. Thank you very much.